Chapter 6 focuses on human genetics, on the inheritance of characteristic in humans, and we will touch upon this feature, why actually it is so difficult to study human diseases and human inheritance. Anyhow, I divided this chapter into two recordings. In the first one, we will discuss techniques which are used to study human genetics, and in the second recording, we will discuss how the information which you generate from these studies, how you can use it in counseling and parental diagnosis. Three important techniques used by geneticists to investigate inheritance in humans are pedigrees, twin studies, and adoption studies. We will discuss each of them individually, but before that, I would like to ask you a question. Why do we need these specialized techniques? Why we can just perform normal genetic crosses? What are the obstacles in studying human inheritance? And if you notice, I already said that, I already answered by my question. Why cannot we do or perform genetic crosses? That's one of the obstacles. It is control matings, which we cannot do in humans. Another one is long generation time, and third one is generally very small number of offsprings we have. Let's go back to matings, control matings. When you perform experimentation following certain traits in model animals, you select parental generation, you cross them, you study F1 generation, you select individuals from F1 which you cross further and to obtain F2 generations. Can you do it with humans? Of course not. We cannot do control mating with humans, and we don't need to go into the detail and reasoning why. You all know why. Now, second obstacle is long generation time. If you work with fruit flies, you can get offsprings in 2 weeks. With the bacteria, it's even faster. Give bacteria optimal conditions and you will have a new generation in 20 minutes. What about humans? You know that we normally have offsprings, we have children when we are 18, 20. So to get F2 generation, it might take anywhere between 35, 40 even years, even longer. It's very difficult to analyze inheritance pattern when you can only follow the outcome after 40 years. And thirdly, we don't have many children. Humans, usually families have 1, 2, rarely 3 children. It's very exceptional that one family has more than 8 or 10 children. So you can imagine to follow inheritance of certain trait and trying to define whether it follows Mendelian inheritance or not, whether it is inherited, whether there is influence on environment, it would be extremely difficult. So let's start with analysis of the 3 techniques, and we will start with pedigrees. So what is pedigree? It is a pictorial representation of family history. When you construct pedigree, you actually are constructing family tree reflecting certain trait which you put in that pictorial representation in the picture using a standard symbol. And all the standard symbols used in pedigrees are outlined in the table on the right, and you will have to learn them. Now, let's start with a basic one. You have to go through all the table and look at every single of the symbols, and you have to learn it. I would just point at certain important points here. Now, square always represents male, and circle always represents female. If this two are unaffected, circle and square, the symbol will be empty. If the symbol depicts a person who is affected, meaning suffers from the disease, that symbol will be filled, like here in the second line. Now, another symbols you have here in the third and fourth lines are symbol for carriers. Be aware that these symbols are used in the pedigrees when geneticists or genetic counsel constructs a pedigree. However, in your quizzes and in your tests, you will not see the symbols. When person or symbol will represent a person who is carrier, you will have to define it. You will have to find out whether that person in pedigree is carrier or not based on the genotype of the parent. So, I repeat again, in your quizzes and on tests, you will not see symbols of carriers. You will have to define carriers by yourself, okay? Now, another thing I want to point out here is proband. So, who is proband? He or she is the first person who comes to the attention of geneticists. Let's say a family has a child with cystic fibrosis and they plan larger families, they want to have more children, but they have a lot of questions. What is the probability that the other child or children might be affected? So they come to geneticists and ask about all these questions. So what the geneticist does first is construct pedigree, and he or she start from the child who is affected because the child was brought to his or her attention first. So that's a proband. Afterwards, he construct pedigree based on this proband, on this child. He includes his parents, his grandparents, his great-grandparents, aunts, uncles, and so on. Now, look on the other symbols on the family. What defines mating? What defines children? Adoption is also defined specifically in pedigree, like here. And if you have, in this case, this is a child which is adopted. So solid line connect the child to his or her biological parents, and dotted line connects her or him to adoptive parents. You have symbol for twins and also for consanguinity or inbreeding. We will discuss this later on, why it is important to define whether that is consanguinity or not. So let's now discuss how do we analyze pedigrees. Before we go there, I want to point your attention on one fact, and it is that it's impossible to define or to see clear Mendelian ratios in single pedigree. Why not? Because the number of offsprings, number of children is too small to see define ratios. So how you can analyze pedigree? You have to learn certain characteristic or certain features which are linked to particular mode of inheritance. And the best way how to do so is to go and analyze some pedigree. So let's start with autosomal recessive traits. There are many diseases which are inherited as autosomal recessive. You have here list of some of them, such as Tay-Sachs disease, galactosemia, cystic fibrosis, or sickle cell anemia, and there are many more of them. So what is characteristics of autosomal recessive traits? Firstly, they appear equally or may appear equally among males and females. There is not clear link to one sex. Secondly, because they are recessive and they can be masked by dominant trait, they can skip several generations. You can have one generation, two or three, you don't see disease, and suddenly reappear in the following one. So they skip generation. And third thing is that autosomal recessive traits are much more frequent in families where close relatives marry, where there is consanguinity. There is increased prevalence of autosomal recessive traits. So how do you analyze these traits? Let's follow this child who is affected in second generation. So she has a disease which, let's label this disease as a disease which is caused by recessive allele A. So she must be homozygous recessive. What do you see? The child can be affected while the parents are not. So other way around, two unaffected parents can have affected child. And how is that possible? This child got one recessive allele from one parent and one from another. So if you follow this mating, you see that each parent has one recessive allele. Okay. However, none of them is affected. What does it tell you? That each of them has also dominant allele, which masks appearance of recessive. So both of these parents are heterozygous. This is how you define genotype of parents based on the phenotype of offspring. Now, normally, if you follow Mendelian inheritance, even if this trait follow Mendelian inheritance, you should have 1/4 of offspring affected, meaning showing the trait. 1/4 would be homozygous recessive. Now, you see you have only 3 children in this family in second generation, so you can even see this ratio properly. Even if you have 6 children, you might not have proper 1/4 of them, or if you have 8 children, you might not have 2 who are affected, because keep in mind that probability 1/4 apply for each birth, for each pregnancy. Okay, so another thing which you have to realize is that if the trait is encoded as autosomal recessive, healthy parents can have affected child. Similarly to autosomal recessive traits, autosomal dominant traits appear equally in males and females. However, they do not skip generations. Usually they do not skip generations, and affected person have always or at least one affected parent. In this case, in second generation, there is affected daughter and affected son, and both got this dominant allele from their mother. Okay, so this is come from mother. Now, I said that they do not usually skip generations. There are certain circumstances under which you have to consider an exception. What I want from you is to look at pedigree and try to look on the possibilities, not just memorize. Autosomal dominant traits do not skip a generation, so if there is not affected child, it cannot be. It can be. When? Let's go over some example. Let's say that this mother here is affected, but she is heterozygous. And of course the father, because he is not affected, is homozygous recessive. So what is the probability that child will be affected? It's 50%. There is 50% probability that the child will carry dominant allele meaning be heterozygous, and there is 50% chance that the child will only carry recessive alleles, so will not be affected. Now imagine scenario where this couple has 2 children or 3 children. There is extreme possibility that none of them will be affected because there is 50% chance that this will happen. Of course, if you have family which has 10 children and none of them is affected while mom is affected, it cannot be autosomal dominant, right? So you always have to think about what it can be and consider all possibilities. Don't just memorize what is said, what probability is. Now let's look what kind of diseases are inherited as autosomal dominant. Achondroplasia, one form of dwarfism. There are different forms of dwarfism, and only one is encoded or inherited as autosomal dominant. Progeria, premature aging, is caused by mutation in lamin A, which is inherited as autosomal dominant. Huntington disease, progressive degeneration of neuronal system. Hypercholesterolemia, which is defect in LDL receptor, and this we will talk a little bit more about this autosomal dominant trait. Familial hypercholesterolemia is a disorder which is labeled in most pedigrees as autosomal dominant, in spite of the fact that it is an incomplete dominance. So why it is? Let's talk about the disorder. We know that cholesterol is not soluble in water, and therefore in order to be transported by blood, it has to bind to proteins and form lipoprotein particles. This might be low density LDL particle, high density HDL particle, or different other particles. In this particular disorder, what happens is that a receptor for LDL for low density lipoproteins is mutating. So what is the outcome? Under normal circumstances, let me go back. Under normal circumstances, LDL receptor binds and internalizes LDL particle. It's a specific receptor on cells binding LDL. Upon internalization, LDL particles are broken down, receptors are recycled, and cholesterol is released and used by cells. You know that cholesterol is important part of cellular membrane, so cell needs cholesterol. So what happened under this disorder? Mutation in LDL receptor. When this mutation takes place, the receptor is non-functional. It cannot internalize LDL particles, and therefore it cannot bring the cholesterol inside cells. Outcome is the cholesterol levels are higher in circulation. Now, there are 2 possible scenarios. Individual can be either heterozygous, so respectively half of the receptors on cells are mutated and half of the receptors are functional. So they still can internalize LDL particles, but only to the certain levels. If the individual is heterozygous, the cholesterol levels reaches twice of normal levels in the bloodstream and leads to coronary artery disorder and potential heart attack. If the person is homozygous for the mutation, so if both receptors are mutated, both alleles are mutated, so there are no functional receptors on the cells, in this case, cell cannot internalize LDL. The bloodstream levels of cholesterol are approximately 6 times higher than in normals and leads to the heart attack as early as in age of 2. And sometimes, if it does not happen so early, it is really inevitable by the age of 20. So we really don't see homozygous for these mutations because it's so deteriorating. So what we observe, respectively, what geneticists observe, what they analyze, what they put in pedigree are usually heterozygous. So it is an intermediate phenotype if you compare it to homozygous. However, that's what we observe because this is so homozygous status is so deteriorating that we don't really encounter such cases. And it's why in most pedigrees you will see this disorder labeled as autosomal dominant. X-linked recessive traits such as red-green color blindness or hemophilia A have a distinct pattern of inheritance. They are usually manifested more frequently in males than in females. And you recall when we discussed sex-linked characteristics and particularly X-linked characteristics, you saw that female woman has to inherit two mutated alleles in order to manifest diseases. Let's look at color blindness. Female has to have both X chromosome carrying mutated allele in order to show color blindness. For example, if she is heterozygous, she would have normal vision. However, since male has only one X chromosome, one mutated allele is sufficient to code for color blindness. So more frequent appearance in males is one of the characteristic of X-linked recessive traits. Now, if the male is one who carries mutated alleles, it is possible that the trait skip generations because if mother is the homozygous wild type, half of the daughter of such couple will be carrier and half will be wild types for both alleles, but none would manifest traits. So under such circumstances, the trait can skip generations. Another important feature of X-linked recessive traits is that the trait is never passed from father to his sons, and it is because father always passes on his son Y chromosomes. He passes X chromosome to his daughter, but always Y chromosome to his sons. X-linked dominant traits usually do not skip generations, and affected males always passes the traits to all his daughters. Look at the pedigree here. The father is affected, so father is XY, where he is affected, so he has mutation on X chromosome. So all his daughter receives this X chromosome, so all of them manifest the traits, while would apply to X-linked recessive applies also here. He never passes a trait to his son because he always gives son Y chromosomes. If affected female is heterozygous, she passes the traits to half of her sons and to half of her daughters. However, if she is homozygous for the X-linked dominant traits, she would pass it to all of her sons and to all of her daughters. Y-linked traits are very easy to recognize because they are always passed from father to his sons, from his sons to his grandson, and so on. So, simplified, all male offspring of unaffected males are affected. So, because of that, these traits do not skip generations, whether they are recessive or dominant, it does not matter, unless the couple has only daughters. In that case, there is no Y-linked traits passed to next generations. As we discussed before, you know that there are very few traits which are encoded on Y chromosome. However, those genes which are linked to maleness are found on Y chromosomes, and they have been shown to be Y-linked. So let's now move on and discuss twins and adoption studies. Both provide invaluable data on the contribution of the genes to phenotypes, to the traits, contrary to potential involvement of the environment. It's really natural experimentation where we do not do experiments, when nature does experimentation and we just observed and recorded. Now let's first talk about twins. As you know, there are two kinds of twins. Either they are monozygotic twins, or we have dizygotic twins. In monozygotic twins, what you know is that one egg, single egg, is fertilized by single sperm and splits very early in during development, and two embryos develop. So basically, these twins, these two children share 100% of genes. In dizygotic twins, basically there are two eggs which are fertilized by two different sperms, so they are the same as, for example, two siblings which are born years apart, and they share on average approximately 50% of genes. So monozygotic twins, 100% of genetic similarities, dizygotic twins approximately 50%. So how do we use this information to study influence of the gene and environment on the inheritance of certain traits? So you look for monozygotic and dizygotic twins, and you look on concordance and discordance. So what is concordance? Let's say you have monozygotic twins. There is one second twins, one twin, second twins, 1, 2, 1, 2. You take these pairs of twins and you look whether they manifest traits you are interested in. If both of them manifest, we said that they are concordant. If only one manifested but second one does not, they are discordant. Okay, so if you go and look how many of these pairs of monozygotic twins manifest the same trait, and basically calculate concordance. Okay, which usually is expressed as percent. Let's say from monozygotic twins, 80% of them, 8 out of 10 pairs manifest the same trait. Now you take dizygotic twins and you do the same. You compare one to both of the siblings and see how many of them manifest the same traits, whether they are concordant or they are discordant. Okay, and calculate percentage. And let's say here you see that there is 40% only which manifest concordance, which are identical, which both manifest the traits, and afterwards you compare it. Now look at the genotype and compare it to concordance. You need to compare both mono and dizygotic. If you have, let's say under this scenario which I've outlined here, situation where monozygotic twins which share 100% of genes, 80% concordance, and dizygotic twins which only share approximately 50% of genes have 40% of the concordance, so only 40% of them manifest the disease. So you see that there is the appearance or manifestation follows the genes. So you say that there is strong genetic involvement in the manifestation of the traits. Now, if you look only to monozygotic twins, if you forget the dizygotic and you look only in monozygotic and you see 100% concordance or 90%, you cannot judge anything, because you might see dizygotic twins where also you will see concordance 90%. What would that mean? That it was the environment in which these twins were raised which affected that traits and not genetic. So you always have to compare manifestation of the traits, meaning concordance in both twins, and compare monozygotic concordance to dizygotic concordance. And if you know the genetic background, it will tell you whether there is genetic predisposition or whether there is environmental factor involved. Let's look in this table. There are several diseases which are outlined. You see heart attack in males. In monozygotic twins, it is 39%. In dizygotic, it is 26%. Since here you have 100% genetic genes identical, here you have approximately only 50% genes identical. And concordance, it's similar, so it does not support genes as the cause of the heart attack. However, let's look in, for example, MS, multiple sclerosis, or even better, epilepsy. In epilepsy, 59% of monozygotic twins are concordant, while only 19% of dizygotic are. So it follows the similarities in genes. However, it is not 100% in monozygotic. What you would say here is that there is some genetic predisposition, so genes play a role in this disease, but there are some environmental factors which also affect, so it's mixed. If contrary, you compare acute infection, you see that both are similar, so there is no genetic link. It's always environment which plays a role. So keep in mind, in twin studies, monozygotic twins have or share 100% of genes, while dizygotic share approximately only 50%. So you have to look in concordance. Concordance means that two twins in one group manifest the same trait. So if concordance is high, let's say if you have concordance in monozygotic twins 100%, it means that all twin pair shows trait. Okay. Afterwards, you calculate concordance within monozygotic twins and within dizygotic twins. Once you have concordance in each group, afterwards you compare them. You compare concordance of monozygotic to concordance of dizygotic. And if this pattern of concordance follows a pattern of genetic similarities, you can say that there is strong genetic background or there is some genetic influence. If both mono and dizygotic twins manifest, let's say 80 or 90% concordance in certain trait, you know that it cannot be genetic, that it is environment. Adoption studies are very powerful tool for distinguishing the effect of genes and environment on certain traits. They were crucial and instrumental in demonstrating that schizophrenia and obesity have certain genetic base and there is certain genetic predisposition. So what adoption studies tell us? If you have adoptee child who is adopted, basically the child shares 50%, approximately 50% of the genes with each of biological parents. However, he or she does not share environment, while the child does not share genes with adoptive parents, but he or she shares 100% of the environment with adoptive parents. So how does it reflect and what does it tell us? If you take such a analysis and you analyze body mass index, for example, in children who were adopted, and compare it to corresponding biological parents and corresponding adoptive parents. So first graph right here shows you comparison of adoptee to biological parents. So you plot BMI of children from scene 1 from low up to high, which you find in obese children, against the corresponding BMI in mothers in red or fathers in blue. And what does it tell you? You can see as BMI in adoptee increases, the BMI of parents increases as well. So there is some correlations, meaning that overweight biological parents tend to have overweight children. So what does it say? It shows that there might be some genetic predisposition. However, let's look to confirm it to comparison of adoptee to their adoptive parents. Here again you have BMI of the children plotted against the corresponding BMI of adoptive mother and father, and you see as you increase, as the BMI of the children increases, there is not real correlation with increase of the adoptive parents' BMI. So what this two graphs together tells you is that there is strong genetic predisposition. So there is genetic predisposition. However, of course, there will be also some environmental factors. If child doesn't have predisposition, but is fed very high fat diet over and over, so he might develop the obesity. So adoption studies really plays important role in defining the potential effect of the genetic and environmental factor on the traits, on certain traits.